So now what we have covered today is that we have done transcription to produce mRNA. The next step is to translate that mRNA into protein, and we will cover that next. Now, as you can imagine, the language of RNA is A, U, C and G. The language of proteins is 20 amino acids. So essentially, a language of DNA or RNA is translated into language of proteins. And this translation process requires a translating table, or like a Rosetta Stone, a genetic code. So because there are only 4 letters, the minimum required word length for these 4 bases turns out to be 64, because if you think of 1, there will be only 4 amino acids. If you think of 2 each, that will be 16 amino acids, right? So this is not possible. This won't also satisfy the requirement for 20. So then we go with 3 nucleotides or all 3 codons, and that produces 64 amino acid combinations, 64 possible combinations. This table here gives the relationship between the 3-letter sequence of RNA and a particular amino acid. Now, if you look at this carefully, what you see is that all the 20 amino acids are listed here, and some more information is also listed here, which will come in a bit. But for some of the amino acids, like leucine is one of the amino acid, there are several 3-letter sequences that code for leucine. Similarly, for isoleucine there are like 3. For leucine there are 6. For serine there is a set of 4. For threonine another set of 4. Alanine 4. Valine 4. Right? Aspartic acid 2. Glutamic acid 2. But there are few which is only 1. Methionine is only 1. AUG, which is also called a start codon, which we'll come to in a bit. Tryptophan is also 1. Then there are 3 sequences, UAA, UAG and UGA, which are called stops signals. That means this is the end point of the transcript. So all transcripts start with AUG, are in multiples of 3, and end with a particular stop codon, which could be either UA, UAG or UGA. All transcripts start with AUG. So therefore, all amino acids sequences or protein sequences start with methionine, and they end with these 3 sequences, which are not encoding for any amino acid, but the protein the sequence of protein will end 1 amino acid before this stop codon. This is called the genetic code. The uniqueness of this genetic code is because of this duplicate or redundant codon sequence for a given amino acid, each species can define which of the sequence codes for which amino acid. what it means is that a bacterial species can code for arginine using this particular sequence CGU, whereas a eukaryotic species can code for arginine using this. This is called codon bias. Certain systems have bias for one sequence versus another. So now that we know how mRNA can be read in multiples of 3 to create a sequence of amino acids, who does the reading? Which protein or a complex of protein is involved in making this process happen? That means reading the RNA code and producing the amino acid chain. And that reading is done by a multiple protein complex and ribosomal RNA called ribosome.